you. I a little bit regret that I didn't talk about my PhD work, which was partially about EGFR, because I'm moving to a completely different subject, but I hope that it will wake you up this time of the day after a very long and exciting day. But I hope it will, because I'm working on a really exciting system, and I do think it's exciting partially because it's different from most systems that we work on in biology. This is not a soluble protein. This is not a membrane protein or transmembrane protein. I work on protein fibrils, and you will be surprised to find that they also have some functional role in some signaling. They are not just structural, but just to define amyloids specifically within protein fibrils, they are very different from other fibrils like collagen or elastin or things like that. But what defines amyloid, they can start with totally disordered species or some native fold that goes partial misfolding that we don't understand why it's happening. And then they start to self-assemble into or soluble oligomers and insoluble fibrils. And what we know about these fibrils is that at least a backbone of the protein is very ordered. Okay, it has a defined structure. The rest of the protein, and this is not drawn to scale because usually the backbone is very small compared to the rest of the protein. It can be totally disordered. It can have some native structure, but it has a structured backbone, and this structured backbone is super stable. This is the main thing that we know about amyloids. And if you ever heard about amyloids, it's probably in the context of neurodegenerative diseases or systemic diseases. There are proteins like amyloid beta and tau that are involved in Alzheimer's, or alpha-synuclein in Parkinson, Huntington in Huntington, ALS, the SOD in ALS, and the prion protein. And the disease is dependent on the type of protein and where it's forming deposits in the brain. Just that in these diseases, I worked on these diseases with David Eisenberg, and it always made me feel a little bit uncomfortable because we don't really understand the etiology of this disease. And as a structural biologist, if you work on things at the angstrom level and you're trying to understand their structure and you don't really understand what's the meaning of this protein in the disease and how exactly they work, you feel kind of detached. But I do want to emphasize that what is very important to know about amyloid is their stability. And I think the best example that you will understand this stability is thinking about prion diseases. So prion proteins, they form amyloid fibrils, so they have all the amyloid characteristics. Some people call them amyloid, some people don't, but it's semantics, but they do have amyloid characteristics. And there were several prion diseases. If it's in sheep or in bovine, they are called the mad cow disease or scrapie in the sheep. And then, if you remember, in the 90s, there was a big, scary event that some people got infected from eating contaminated meat. They started in England, that the cows were sick, and then people eating from the sick cow got a disease that was called, or a variation of it, a Creutzfeldt-Jakob disease, which is a really horrible neurodegenerative disease. The first prion disease was actually found in a tribe in Papua New Guinea of cannibals, that they were eating people's meat, including the brain. So they got this prion protein from the contaminated meat of their friends. But why I'm talking about these diseases in relation to amyloids is because they explain the stability of the amyloid. Think about, this is an infectious disease, okay? What I mean, it's not really infectious disease. I don't know if I'm allowed to call it like that, but it's a transmissible disease. You get it from eating contaminated meat, okay? And if we are used to think about transmissible diseases, we are used to think that a microorganism is involved. But here, no microorganisms are involved. It's no virus. It's not a bacteria or a fungi. It's a protein. So this disease is being transmissible by proteins. And the reason that it's even possible, because we are talking about eating something, we are not talking about injecting something. You can also inject amyloid beta and get Alzheimer's, but here I'm talking about eating. So these proteins are so super stable that they can go all through the digestive system, all the proteases, the low pH in the stomach, all the microorganisms. They can go into the blood and find their way to the brain and catch up with other protein and pull them in to form this amyloid deposits and which lead to neurodegenerative diseases. So there are special proteins, you have to admit. They're not they do have signaling issues that I will mention later, but you have to appreciate at least their phenomenal stability. So if we want to understand the structures of amyloid, this was super challenging, at least for crystallography, because if we are talking about fibrils that are ordered, but they have an ordered spine and the rest of the protein is completely disordered, so how do you work on something like that with crystallography? So the method that was developed by David Eisenberg was to identify the structured region in the fibril that form the backbone of the amyloid, and then determine the structure of this fibril. There is a lot of debate in the field about if this approach is relevant or not, but David and others, Udi Gazit, showed that really few residues, even 4 residues. Udi Gazit went to 1 residue. David Eisenberg usually worked with 6 residues. They showed that they can recapitulate all the biophysical properties of the full-length protein, and David also showed that you can design drugs based on the structures of these things, and they affect the full-length protein. So even if you are not totally convinced that the structures of short segments are totally reflecting what happens in the full length, you can at least appreciate that they provide us with a very reasonable model. And the best things about working with spines of amyloid is that they form crystals and we can determine the structure. Just that these crystals are super tiny and they are fibril-like, they are like needles, they are very long. It's very hard to determine their structure. The methodology is unique. All the way from crystal mounting to solving the structure. It's not something that a crystallographer, a trained crystallographer would easily do. It's something that you need to specifically learn, and luckily I did this with David. And they solved the first structure more than a decade ago, and the motif in all of these fiber-like structures was a pair of beta sheets formed from segments stuck in register. So on the right, you're looking down the fibril axis, and you can see amino acid protruding from the sheets, forming this dry, self-complementing interface bonding the sheet, and David called it a steric zipper, because think about it, you have 2 sheets with side chain interdigitating like that, and then it goes all the way to the fibril axis. It can be hundreds of thousands of layers like that. And so they resemble a zipper, okay? So it creates like a serious zipper. And this structure manifests the self-seating characteristic of the amyloids and their high stability. You can understand why this structure is super stable. And since then, David solved the structure of over 200 of the steric zipper from more than a dozen disease-related proteins, and the vast majority of them are indeed steric zippers. Crystallography is not the only method to determine structures of amyloid. I started with that because this is what we do. But there was a lot of information coming mostly from solid state NMR, from Tikos and Roland Rick's lab and Beata Meyer. They work on amyloid beta involved in Alzheimer's and determine structures, or this is a part of the East Prion protein. And micro-ED by Tamir Gonen recently came to light to solve structure from almost invisible crystals. And obviously the resolution revolution of cryo-EM didn't skip amyloid, and just in the last year 2 structures from cryo-EM at high resolution were solved of the amyloid beta and the tau proteins, both involved in Alzheimer's. But all of these structures, like the crystal structure of David, showed that the amyloid have specific type of form that was called a cross beta fibril. So this is how you differentiate amyloid fibrils from all other kind of fibrils. They form cross beta fibrils. it means 2 mating sheets, and the cross is coming from that each subunit, each strand is perpendicular to the fibril axis, so it forms a cross. Okay, so the strands are orthogonal to the fibril. And this is how amyloids are defined, basically until today. Nomenclature is an issue. I will talk about it later. But they are cross beta fibrils. And even in the 70s, the 80s, people studied amyloid with fibril diffraction. So they shoot X-rays on the fibrils, and they indeed saw a pattern that kept on appearing. And now we understand this pattern that was it is a cross beta pattern because you have a diffraction ring at 4.7 angstroms, which is the distance between strands on a sheet, and it's orthogonal to the distance between sheets. Okay, so if you want today to define an amyloid, you take fibril diffraction. If you get a cross beta, you are allowed to call it amyloid. But now I don't want to talk at all about amyloids related to neurodegenerative diseases or to systemic disease. I want to move to a completely different system, which for me is very exciting. I think it's very diverse. It's very different than human amyloids, but there are some commonalities and differences. So about a decade ago, Matt Chapman from Michigan discovered that fibrils secreted by E. coli and Salmonella, called Corley fibrils, they have similar biophysical characteristics as human amyloids, and they characterize them. And these fibrils are involved in the structuring of the bacterial biofilm. And if you are not familiar with biofilm, I presented this late evening thing before dinner. This yucky thing. So basically, it's a yucky agglomeration of bacteria. So a bunch of bacteria assemble together. They secrete sugars and proteins and DNA and whatnot, and they sit in this extracellular matrix that they secrete. And the biofilm helps the bacteria or the fungi attach to surfaces like tissues, and it protects it from the environment. So you cannot treat something like this with antibiotics. It doesn't penetrate. You cannot treat it even with solvents. They don't go across the biofilm. If you have an infection that involves biofilm on the skin, you have to surgically remove it. You cannot treat it with antibiotics. If you have biofilm on implants, you have to take the implants out. You cannot treat them. The same with teeth, and the same with environmental. Think of what happens in the sewage beneath the hospitals with all the bacteria of the hospital and antibiotics of the hospital go into these pipes. So what kind of bacteria and biofilm are happening there? And amyloids are an important constituent of this biofilm because they make it super, super ultra stable. I talked a lot about the stability of the amyloids, and the reason what I wanted to go into is that you can understand that they stabilize the biofilm. I imagine it like a tennis rack that the bacteria sits on something that is fibrillar, and it doesn't let anything get through. So you add proteases, solvents, whatever, it doesn't kill the amyloid, so the biofilm sustains. So this is one role of the amyloids, but this is not the only one. They help with adhesion to surfaces in tissues, in changing surface properties, like protecting spores from water. And they are affecting the host, and they are involved in many signaling pathways, especially in the immune system. So they are very tricky and manipulative. They can trigger the immune system. They can actually inhibit the immune system in a way that it will not attack the bacteria. And they do induce some pro-inflammatory signaling, and they can kill human blood cells to protect themselves. So they kill the erythrocyte and the immune cells. And they do really many sort of things. They're super, super smart. And they also can kill other bacteria if they want to spread over some kind of niche. And when you think about all these properties of bacterial amyloid, microbial amyloid, they were studied in the last 15 years, but there were no structural information available for bacterial amyloid. The only microbial information is the yeast prion, but the yeast prion are kind of like human pathologies. So there is no information about microbial functional amyloids. And when I looked at that, I thought, does it make sense that this cross-beta structure of the human pathological amyloid can participate in all of these different functions? Like, it sounds impossible, right? How a cross-beta stable fiber can do all of these things. Does it make sense? So about 5 years ago, I decided that my lab will deal with microbial amyloids, and we need to study their structure and their structure-function relationship, and this is what we did, and I want to share some stories that we got. So the first thing that we noticed, that every time that we work on amyloids from bacteria that is involved in biofilm, and we determine structure of segments from this amyloid, we get the canonical steric zipper structure of pathological amyloid. So on one hand, it was not surprising. On the other hand, it was still cool because it's the first time that we determine structure of amyloid from bacteria, and you can see the same things that you saw with human amyloids. And it makes a lot of sense because I told you for so many times that amyloids are super stable. So if you're involved in biofilm, you will take the stability of the human amyloids and use it for the biofilm. And then we thought, okay, if biofilm-related proteins have similar structures as human amyloids, and there were so many drugs developed against human amyloids, and they don't work in the clinic because we don't understand Alzheimer's, but here we understand what amyloids are doing, so let's import some of these compounds and test them on the biofilm-related amyloid. And this is what we did. So we started with collaborating with Dieter Wilbord from Jülich in Germany. He designed it's the D-amino acid peptides. So they are peptides, relatively short peptides. They are composed of D-amino acids for bioavailability. And they developed these peptides against amyloid beta involved in Alzheimer's. And some of these D peptides are actually in clinical trials because they show cognitive improvement in mice. But we took these D peptides and we said, okay, we think we are very similar to amyloid beta. And we determined structure from the CSGA protein. It's amyloid secreted by E. coli and Salmonella. And we did a series of experiments. So several students were involved. Sergey started it, and Nimrod and Nir jumped in because we wanted to use a lot of different, like it's the first time that we show something like this about amyloid. So we wanted to use a lot of different methodologies. So we monitored kinetics of amyloid formation with amyloid-specific dyes and showed that the D peptides do inhibit the bacterial protein. We monitor structural changes with CD and electron microscopy. And my favorite thing is that this amyloid is so stable that if you boil it in 95 degrees in SDS for 10 minutes, it's still, the fibrils, they don't disassemble. They're still insoluble. So we can use it for assays because you can produce the protein. It migrates on a gel when it's fresh. But after like a couple of hours, it stopped migrating on the gel because it's insoluble anymore, and it's insoluble in SDS. But if we add our drugs, our dipeptides, then we can see that they still can migrate on a gel, meaning that they're still soluble and they were delayed in fibril formation. I like this experiment because it's super elegant and it doesn't require dye binding or any other properties. It's just a clean way of looking on soluble versus insoluble. So we found two dipeptides that were designed against amyloid beta that work beautifully on inhibiting our CSGA protein from the biofilm of E. coli and Salmonella. And the most exciting things afterwards, that it actually worked in vivo. So Ophir grew a biofilm of Salmonella. He added these two compounds. Here I'm showing only one. And 10 micromolar of the dipeptides are enough to significantly reduce the biofilm formation of Salmonella. So it's
before it's made or after it's
before. Yeah. You cannot kill amyloids after they are formed. This is, I think, the problem with Alzheimer's as well. Even if the oligomers are, you cannot kill amyloid. People claim that EGCG from green tea disassemble amyloids, and there are several compounds, but it's super. You cannot kill it with guanidinium at 10 molar, like.
So you can prevent the formation.
Yes. So the idea, but with this kind of like biofilm, for example, is a huge problem on catheters. So if you have compounds that prevent biofilm formation, this is applicative. This is something that you can do beforehand. You can put it on implants. Once the biofilm is formed, you're pretty much, yep. So they prevent biofilm formation of salmonella, which is pretty cool. It's cool in different ways, because it's not just that we found an anti-biofilm drug that is currently unavailable. It's the idea that you import something that was designed for Alzheimer's, and just based on the insight of structural similarity between bacteria and human amyloids, you can import them to do something else, like being anti-biofilm drugs. And it tells us that we now can try so many different compounds that were designed for Alzheimer's, Parkinson's, whatever. I'm not talking about cholinergic approaches, but anti-amyloid approaches, and use them and try them as anti-biofilm drugs against various kinds of bacteria. So this is what we are trying to do now. So this was the salmonella story. Now I want to move to a really cool, stop saying cool, Natalie. She, okay, I'll try. Can I do awesome? Okay, so it's, I like the system because I have a personal attachment to Staph aureus. It's a nasty bacteria. I didn't walk for 2 years during high school because of Staph aureus, so I have a personal thing with it. So Staph is nasty, and there are some strains that are totally resistant to antibiotics. And Staph aureus secretes a family of peptides called phenol-soluble modulins, or PSMs in short. They are relatively short peptides. And in some virulent strains, 60% of the protein biomass that is being secreted by the bacteria are PSMs, so are definitely important to the pathology, to the infections caused by this bacteria. And they are indeed involved in a lot of virulent activities, like surface spreading, biofilm again, antimicrobial activity against competing bacteria and microbe. Their cytolytic activity, so they kill human cells, especially the immune system and blood cells, and they produce some pro-inflammatory activity. And a few years ago, the lab of Blaise Boulez suggested that PSMs have some amyloidogenic properties, but it wasn't investigated in terms much of a structure-function relationship, but there were really nice indication by Blaise that about this amyloid tendency. And I'll start with the biofilm role because I already talked about that. So Nir in the lab worked on 2 PSMs, PSM alpha 1 and alpha 4, that we already knew that they are involved in the biofilm of the Staph aureus. And they indeed form amyloid-like fibrils, and their fibril diffraction pattern show the canonical cross-beta pattern of human amyloids. So this is just another example, like the salmonella, that if you are involved in biofilm, even if you are a short peptide, this is just 20 amino acid peptides, you can form the canonical amyloid conformation. We wanted to get atomic resolution information. Even with 20 amino acids, they are too long to see structures because some of it is disordered. So we identified the amyloidogenic spines of these peptides. We also did mutations to confirm. We use computational approach, but then we do mutation to really confirm that we have the spine of the amyloid. And we determined the crystal structures, and surprising or not surprising, it was a steric zipper, as we expected, that corresponds to the fibril diffraction pattern of the full-length peptide, the cross-beta pattern. So until now, I didn't tell you anything new. Another example of biofilm and steric zipper conformation. I just want to mention that what is really interesting about PSMs is that they are being truncated in vivo to active derivatives. So the stuff doesn't secrete the PSM as they are. There are proteases that truncate the peptide, and then you have derivative with gain of function. So it's super smart and super effective. And we indeed see that in all of these truncations, the spines that we predicted sustain. So it's something fundamental probably in the sequence. Okay, so then we moved to another PSM that there was no indication that it even formed amyloids. And we said, okay, let's look on the equivalent segment that we also predicted to be amyloidogenic in PSM alpha 3. Okay, PSM alpha 3 is not involved in biofilm. Okay, it's mostly cytolytic. And when we started working on this segment, we discovered that this segment and the one residue longer segment, the seven residue segment, they have antibacterial activity. Okay, and it's active against Micrococcus luteus and Staphylococcus suminis. And why did we even look on antibacterial activity? Because recently it was suggested by Moyer that amyloid beta involved in Alzheimer's has some antibacterial activity, and people then showed that for alpha-synuclein and other human amyloids. And there was a suggestion that people in the field don't really like, but it's still out there, that maybe the physiological role of this amyloid beta and alpha-synuclein, that we still don't know what is the physiological role. But maybe they have antibacterial activity, and in some point of human life, the brain was threatened by this by bacterial infections, and then it produced amyloid beta. And then 30 years later, when we are not really supposed to live that long, because then the same person got Alzheimer's. So there is a lot of, for me at least, I think it's very interesting to understand what's the role of microbial infections, and not just pathogens, also the microbiome in correlation to neurodegenerative diseases. There are a lot of correlative kind of information that suggests that there is correlation between bacteria and neurodegenerative diseases, but nobody did a molecular level investigation or something like this, which is something that we're very interesting to look at. So all this antibacterial activity came up, and people also showed that some known antibacterial peptides form amyloids. So we tested antibacterial activity, and indeed it worked. But what was interesting is that it wasn't active against Staph aureus, the secreting bacteria. So the secreting bacteria knows how to protect itself from its own peptide. And then we thought, okay, what makes it antibacterial? Is it the fact that these segments form fibrils? Maybe it's the fact that they have hydrophobic and positively charged residues, okay? But the equivalent segments that I talked about before from PSM alpha 1 and alpha 4 that are involved in biofilm also form fibrils, and they also have hydrophobic and positively charged residues, but they are not antibacterial, okay? So it's not just the mere sequence property or the ability to form fibrils. So we thought maybe it related to the architecture of the fibrils that they form. And we determined the structure. Indeed, we found something new and interesting. It forms a fibril, but now you're looking down the fibril, and instead of mated beta sheets, you have beta sheets that are arranged in kind of a hexameric configuration with a sub-nanochannel in the middle, okay? So it's a new type of amyloid configuration. We never saw something like this before. It's amyloid because it's composed of elongated beta sheets. You have fibrils. You can also see them in EM. But another configuration, so I cannot promise you that this configuration is encoding the biofilm, the antibacterial activity, but I'm telling you this is the structure. It has antibacterial activity. Other peptide that has a different structure does not have this activity. So I think even if we don't understand exactly what's going on, we see that there are differences. And I mentioned stability again and again. And if we look on thermostability of fibrils, so if we take the ones that form the canonical steric zipper structures, they are highly stable also when you elevate the temperature. Okay, so in 50 degrees, they are still stable. But this guy that forms this atypical crystal structure is not stable anymore. Okay, so when you elevate the temperature, the fibrils disassemble. When you bring it back to room temperature, the fibrils form again. So you have something that this one has a regulation that this one doesn't have. This is a structural stability issue, and this one has lack of structural stability. So it fits with the structural properties that we see. We also did FTIR and CD on the fibrils, and we indeed see that the differences in structures that we see in crystals corresponds to the differences that we see in fibrils. So we have a really beautiful structure-function relationship here when this guy involved in biofilm forms the cross-beta structure that has a structural role, and this guy with atypical structure has the antibacterial role. So this was the story with the spines, but then I didn't tell you anything about the full-length PSM alpha 3. I just told you a segment of it. But we wanted to investigate if PSM alpha 3, the full-length, even has like these amyloid properties. We didn't know. What we did know about this peptide, it's the most cytotoxic member of the family. And this is, so we already knew that. And I'm going to show you a work done by Einav Taif-Fligerman, who left for a postdoc a few months ago. I'm still crying since then. I hope that she will succeed. So she showed that PSM alpha 3 can indeed form these beautiful fibrils, and they bind the amyloid indicator dye, tau-flavin T. They even bind the dye better than amyloid beta involved in Alzheimer. You can see the fibrils in confocal microscopy. We can never get these beautiful images with human amyloids, which is nice. And with normal amyloid, you expect that they will undergo structural transition from whatever, unstructured, helical, whatever, to some kind of a beta-rich structure. But when we look on secondary structure properties in solution by CD, but also in fibrils using FTIR or solid-state CD, you can see that PSM alpha 1 is helical in solution and in fibrils. So what is going on? Because if you are an amyloid, you are expected to be beta-rich. So, but now it doesn't happen. And we did this experiment thousands of times. They never changed their structural properties into beta. And then crystallography kind of solved this paradox. And when we solved the crystal structure, I was completely amazed from what I'm looking at. Actually, I want to be honest. We didn't know about the helical properties only after we solved the crystal structure, okay? We just like sometimes you just try crazy things, and we never tried to crystallize amyloids that are that long, but we said, whatever, let's try to crystallize it. And crystals grew, and they were amyloid-like in properties, and I thought, wow, a full-length amyloid. This will be the first full-length amyloid to ever be crystallized. And the diffraction was horrible. It required 7 synchrotron trips and like many months of working on these crystals, but we didn't want to give up because it's amyloid crystals, and this is the first full length. Back then it was the first bacterial amyloids to get that we got crystals on. And when we finally solved the structure, in crystallography, you're looking on what happens in the asymmetric unit. You don't look on the self-assembly at the first sight. And I solved the structure, and I saw a helix. And I like, shit, I started cursing like hell because I worked for so many months to get a monomeric structure, okay? What's a helix? I knew you are a helix as a monomer, but you know. So I was very upset, and I went to do something else, writing a grant. And then I got bored from writing the grant and said, okay, let's look on the structure. And then I looked on the crystal, the assembly of the helices, and then it hit me what I'm looking at, that I'm actually looking on a cross-alpha structure instead of a cross-beta structure. So you have 2 mating sheets exactly as in other amyloids, and they form this dry and tight interface. But the individual units are not beta strands, they're alpha helices. So we call it a cross-alpha structure, and it was very exciting at the time because we understood that on top of providing the first crystal structure of a bacterial amyloid and the first crystal structure ever of a full-length amyloid, we see something that is unique in protein science, right? Mating sheets of alpha helices is not something that you see every day. And we hopefully extend the definition of amyloids being not just cross beta, being also cross alpha. I still get a lot of rejection from several scientists in the field that they don't allow me to call it amyloid, but then I'll tell them if I don't call it amyloid, to what conferences I'm going to go, okay? Like, who will take me? So I need to call it amyloid at least, amyloid-like if you wish. So this was the structure, and it's not this kind of cross alpha doesn't appear only in the crystal. It's also appear in the fibrils themselves. The diffraction pattern is a cross alpha, and as much as you are always excited when you get when you find the fundamental when you get fundamental insight like solving a crystal structure, but people don't realize that sometimes going to a synchrotron and getting fibril diffraction can really make you feel happy, because I'm looking on cross beta diffractions for several years, and to see a cross alpha diffraction in fibrils was I really didn't believe that we are going to see it, but we did. So in some points, the little things can be more exciting than the big things. So if you compare a canonical cross beta structure to the cross alpha structure, you can realize that they are very similar on the quaternary structure level, but they are very different in the secondary structure level. So it's a unique kind of polymorphism. It's a secondary structure polymorphism. And then we wanted to know, science reviewers wanted to know what's the functional role of this cross alpha fibrils, because, okay, maybe it's just a cool structure, but nothing more than that. How do you know that it's functional or not? So we worked for many months to try to, and we eventually succeeded with mutants and other means. I don't want to get into that. But the endpoint is that fibrillation enhances toxicity. So if you remain amphipatic helix, but like what single point mutation can sustain the helix, but it prevents fibril formation, so you're not toxic anymore. And we have tons of mutations and tons of data right now that I can say it with no hesitation. Fibrillation is needed for the toxicity, and here the toxicity was tested against human T cells, like human blood cells, and we showed it on HEK cells. So toxicity is not a specific mechanism. It kills all types of cells, and it kills it really fast and in low concentration. About 2 micromolars of these peptides are enough to create massive cell death. So, so cross-alpha fibrils are functional amyloids that work in order they participate in infection. So the staph aureus in the blood, it secretes the PSM alpha 3, it kills the immune system, and this is the mechanism. And in collaboration with Raz Jelinek and Ravit Melishev from Ben Gurion University, they did a series of biophysical experiments using membrane mimetics and with a series of mutations that we have. And they showed that the fibrils themselves are interacting not with the surface of the membrane, but with the hydrophobic part of the membrane. So we don't really know how they are being inserted, but if you keep it monomeric, it doesn't go into the inner part of the membrane. So the mechanism of toxicity is related to membrane deformation. And the reason that we know that it's not a protein-dependent mechanism is that we took the sequence and we synthesized the peptide with all the amino acids, okay? So all the amino acid is still toxic as much as the wild type. It forms fibrils as much as the wild type, but so it's still toxic to the membrane, and it's still interacting with the membrane the same way. So if it's the amino acid, that you know that stereospecificity is really crucial for interaction with protein, but it's not crucial for interactions with lipids. So the fact that it forms fibrils, that it forms cross-alpha fibrils, and it's toxic, tells us that it's protein-independent toxicity that probably comes from membrane deformation. And these are confocal microscopy images and SEM images that Einav took. And 5 minutes after incubation with, 10 micromolars of this peptide, the T cells, you can look at graveyard of T cells. Okay, so after 5 minutes, it totally kills the cell. It's a highly toxic peptide, and you can I have now better confocal images. You can see the aggregation of the peptide on the membrane, and you can see that in spots when you started seeing aggregation on the membrane, you see membrane deformation and the cell dies. So it's a protein-independent membrane deformation cell toxicity. So think about these tough hours and the crazy family of PSMs, because just by secreting 3 peptides, and I want to remind you that they are being truncated in vivo. So you have a cross-beta structure for biofilm structuring. You have this cross-alpha for cytotoxicity. You have these derivatives for antibacterial activity. So you get a lot of virulent activity just by secreting one family, and we didn't work on most of the PSM by now, so who knows what we are going to find. And if you're asking yourself about a cross-alpha, is the cross-alpha a one-time thing? Is the only time in nature that you will find such a structure? So it's not a one-time thing. People didn't understand what they are looking at and didn't look on this type of things, and they are very hard to solve technically. The crystal structure is very difficult to solve. But what now we know that there are more. And one example is an antibacterial peptide that is being secreted by frogs. So it's eukaryotic peptide, and it also formed this cross-alpha type of structures. This is amphipathic helices, like the previous one, that forms these rows of mating sheets. This frog peptide, it forms the fibrils, it binds the amyloid indicator dye. And what's nice about this peptide is that it's basically unstructured when it's monomeric, but if you add lipids like membranes, it's moving to be helical immediately. So the membrane of the bacteria affect the peptide to be more helical, probably enhancing fibrillation. And if you incubate the soluble peptide with bacteria, even at 4 micromolars overnight, you can already see fibrils covering the bacteria and killing it. So there is a kind of bidirectional relationship. This is true also the PSM alpha 3 with human cells, and here with bacterial cells. So the cells are inducing fibrillation. This is what we know now. And then the fibrils kill the cell, which is really interesting. And I want to finish by saying that amyloids are not the only cool fibrils out there. Because we started looking more and more on the relation between antibacterial activity and self-assembly, we came across a human-derived antibacterial peptide, which I don't want to say its name. Solving the crystal structure after so many attempts, it was worth it, because we discovered something really novel. I think there is a novel type of fibrils that we didn't see before. It's composed of 4 helical bundles that have hydrophobic core, and they are arranged in this hexameric configuration, elongated configuration, as you can see that. It looks like a decoration of something, right? And in the middle, you have a sub-nanochannels that is flanked by lysine residues. It's big enough to hold amino acids, ions, whatever that we are going to test that. So we only got the crystal structure and a few indications that this one interacts with amyloid beta, so there's a lot of communication and really cool stuff. But
what does this peptide do?
It's in the body, it's antibacterial.
Antibacterial.
Yes.
Presumably pokes holes in bacterial membranes.
Yeah, so we don't know. So what we want to test, if you put it in some reconstitution in liposome, if it really can. Yeah, but there is another thing that we are looking at on interactions with human amyloids with immunomodulation. So there are so many, like it starts so many different fields, like immunomodulation, neurodegenerative diseases, antibacterial activity and mechanism, like blah, blah, blah. Like I have 3 students.
Why is it selected to bacteria?
It's a good question, but most of them are not truly selective. Like if it's a concentration, but most of them can also be cytotoxic. But you know what's weird? If you change it to D-amino acids, they stay antibacterial, but they are not cytotoxic anymore. So the mechanism is not the same mechanism. So so this is what it is, and I hope that you.
Want to say what to keep because you want to make money?
No, I'm not. Sorry. No, just because I don't want to be scooped. Yeah, no, I'm not. Yeah, I'm so not there to think about industry. I'm so not there, but I should be. No, just scooping. You know, it's a novel type of fibrils. I'm a small lab, Israel, I just don't want to be scooped. So I hope that even if it's not signaling, you appreciated microbial amyloids. And the implication for such work is obviously in idea to design new antibacterial drugs, because the amyloids are the targets for the antibacterial drugs and antibacterial drugs themselves. Also, biotechnology-wise, there are materials for novel technologies. I think they participate in communication between microbes and communication between the host and the microbes, as I discussed. So all of these things that we want to look at. And this is my beautiful lab back at the Technion. And thank you, all of you, and the funding. And thank you for inviting me to this beautiful conference. And